Okay, hello everyone, welcome to this video in the Biotech Basics playlist. This is the first video, as you can probably see, with a new level of technology. So we've gone full green screen in here. So I'm going to now move directly to within my presentation there. So this is the first of these videos I'm doing this year in 2021, and this is going to go in with the Biotech Basics playlist to add to that, which were actually some of the first videos I did when the pandemic started last year. So this playlist is going to be somewhat jarring from the grainy picture-in-picture when I first started right now to all the way through to full green screen. It's like the finest visual effects of 1998. So today we're going to basically look at recombinant DNA technology. Now with this topic I always think there's not really a good way in, so you just kind of have to start somewhere, because every technique relies on a whole bunch of other techniques. As you'll see when you get towards the end of this topic, they are all interlinked, they're all intertwined, and you can't be certain of what is going to be necessary for each individual technique. Okay, so there's going to be lots of gaps. I'm sure a load of you will spot in this and go, okay, but how does that work? Well, and great, right? Good. I'm glad you're spotting those things, but you just have to take it from me that there are going to be other videos that fill in all those gaps and explain all those extra bits and bobs. But so today we're going to start with recombinant DNA technology, the idea of genetic modification. So genetic modification, there it is, producing DNA fragments, and I will get the gestures right at some point. Still fairly new technology for me. So here's the bit of syllabus. We've got to basically get a definition of what recombinant DNA technology is, the idea of involving transferring DNA from one organism into another, and this only works because the genetic code is universal. We're then also going to talk about how we generate DNA fragments. The idea of, if you want to do this, you've got to take a bit of DNA from somewhere to then insert into your host organism. And there's three different ways of doing this. You've got conversion of mRNA into complementary DNA. You've got the use of restriction enzymes to cut the DNA out of the genome directly. And you've got, just starting from scratch, completely generating an artificial DNA strand based on the sequence of maybe the amino acids in a protein. Okay, that's called creating a new gene in a gene machine. Okay, so this is all very, very modern stuff. So what do we mean when we say genetic engineering, and why is it such a hot topic and has been for years now? Why are we so interested in genetic engineering? Well, basically it's about inserting foreign genetic information into a new organism. Okay, so it generates what we call recombinant organisms. For example, you've got bacteria that produce insulin, that's very routine now. You've got plants that are resistant to certain herbicides, so you can get rid of weeds, or resistant to certain pests, so you have to use less pesticide on them. And then of course you've got the sort of headline grabbing, slightly more exciting examples like glow-in-the-dark mice, which is really just a proof of concept. Can't really think of a use for a glow-in-the-dark mouse, apart from, you know, making it easier for cats as long as they've got UV lamps. So there's that, and then there's of course things like spider goat, which has had a lot of traction recently. The idea of a goat producing spider silk in its milk that can then be used in medical implantations and so on and so forth. Okay, so those are sort of the more high-profile versions. Okay. So this is essentially the molecular biologist's toolbox. Okay, it's a series of tools and techniques that every molecular biologist will need to know, regardless of what area of biology they're in, from forensics to anthropology to manufacture of vaccines to looking into genetic diseases and medicine, cancer research. These are all absolute bread and butter tools that your molecular modern biologist has to know about, has to use. So we've got here, look, a little list. So we've got our restriction enzymes, our DNA ligase, our vectors and plasmids, our gene transfer, our genetic markers, our polymerase chain reaction, our DNA probes, our gene synthesis, and our electrophoresis. And we're really going to focus on restriction enzymes, ligase, and gene synthesis in this video. So moving straight on. What are the stages that you need to undergo in broad terms to do recombinant DNA technology? Well, first thing, you need to isolate DNA fragments which contain the desired gene. Okay, so you've got to find the DNA code in the first place. If you've got a cell that's doing something cool, you're looking at a jellyfish going, well, that's glowing in the dark, or it's shimmering in a nice way. I wonder if that's to do with a protein. You've then got to find what gene codes for that protein. Okay, and there are a number of ways to do that. Once you've isolated that bit of DNA, you've then got to take it and insert it into a form that your host organism is going to understand. We call that a vector. Okay, most common one of those probably is a plasmid, so a circle of DNA that works in bacteria. So that is a plasmid, and the bacteria will then understand that this is a gene it's supposed to transcribe and translate and manufacture a protein from. Whereas if you just chuck the DNA in there, it's just going to get broken down. Okay. Once you've got your nice made vector that the bacteria or other cell will understand, you've then got to somehow get that in there. You've got to actually get it into the cell, which is called transformation. Now that's quite straightforward in single cells on a dish, in bacteria on a dish, but if you are doing this from a medical perspective and you're trying to use, say, a gene therapy, the problem you've got with a patient in front of you in the clinic is a patient is a pile of billions of cells. How do you get the new gene into every single one of those cells so as to stop the disease process? Okay, and maybe you don't need to get it into every single cell, maybe you need to get it into some cell, but that transformation process is absolutely critical. After that, you need some sort of identification. Okay, so how do you know that it worked? Because obviously you've got cells, you've done a thing, you can't see that the gene has gone in, unless of course there's something to mark out the cells where this has worked. Do they glow in the dark? Are they resistant to antibiotics? Something like that, something visually obvious that you can see to find the ones that have worked and get rid of the ones that haven't. Okay, and then once you've got the ones that have worked, you've then got a cloning process where you're going to actually hopefully grow those cells in a dish, or take that into the clinic, put that in a person, and if it is growing cells in a dish, then you're going to harvest your protein product off. So with insulin, you're harvesting the protein product from a big fermenter of these bacteria that are producing that protein for you. Okay, so the reason this works, the only reason this works, is because the genetic code is universal. Okay, it doesn't matter whether you're virus, bacteria, animal, plant, your A's, T's, C's, G's, whatever, code for the same thing again and again in any species. Okay, so A's, T's, C's and G's mean the same in bacteria, viruses, fungi, us, doesn't matter. Okay, you've also got then obviously the amino acids are coded for by the same amino acid, same genetic code, whatever. So if you take that DNA and you place it into another organism, right, it will produce the same protein. That's the only reason genetic engineering works. And if you're not sure on why this is the case or how transcription translation works, then look back to previous playlists on protein production. They're also on the channel. So where do you get this DNA from? I mentioned there's a number of places you can get these DNA fragments from. Well, let's go through those to start with, right? So first place is using the mRNA. Now this is probably the smartest way to do it. If you've got a cell that's manufacturing a protein that you like, okay, then of course you've got some cells that aren't. It stands to reason that this cell is going to be producing, these cells are going to be producing an mRNA to code for that protein, where these cells over here are not. Okay, so you look for the mRNA, you isolate the mRNA, and then you have to work backwards from that to get DNA, or what's called cloned or copied DNA. You can go direct into the genome and start cutting the DNA directly out of the genome, but as we know, if this is a eukaryote we're talking about, the genome is massive, and only 1 to 2% of the DNA actually codes directly for protein. So not is it looking for a needle in a haystack, it's looking for a needle in a few billion haystacks. And because in us mRNA has to go through splicing to become mature mRNA, that DNA will still have its introns in there. So you're not only looking for a needle, but you're looking for a needle that's been cut up and been interspersed with even more hay. Okay, so it's really, really difficult to do. Okay. Or you could just forget all this. You could just go the high-tech route, the modern, really, really modern route, and go, well, we could just look at the protein. We could just sequence the protein and work out what amino acids we need in what order, and then work backward from that to the RNA, to the DNA, and just design that on a computer and print it out using a DNA printer. Right? That's more and more common. Or, even better, download the sequence of that protein from somebody else who's sequenced it already and uploaded the sequence onto a database somewhere. Okay, that's the gene machine process. So let's go through them one at a time. First one is going backwards from the RNA into DNA. Okay, this so-called reversing transcriptase, reversing transcription. Okay, and nature will provide, if you look hard enough, there's usually an enzyme or protein out there that will do something useful, and this is no exception. Reverse transcriptase is an enzyme that does exactly this. Now, the only organisms in life that are likely to have RNA and need in nature to go back to DNA are viruses, or so-called retroviruses. The retro bit literally means going backwards. Okay, So our retroviruses, like HIV, which have an RNA genome, need, when they infect your cells, to go back to DNA such that they can cause your cells to produce more RNA for them. Okay, so they use reverse transcriptase to do so, and we've actually hijacked that for our own purposes. Okay, what you manufacture from that is a double-stranded molecule of DNA that is the DNA equivalent of that RNA, or so-called cDNA, cloned DNA, copied DNA. Right, this here is a diagram of how that works. Initially, you've got your RNA template up here. Right, this then goes through polymerization. Right, so the DNA is formed along here. Okay, using a sort of hybrid with the DNA and the RNA, and then eventually the RNA is got rid of. Okay, and you manufacture using polymerase enzyme, just like in DNA replication. You manufacture the other strand, and you end up with a double-stranded DNA molecule. Okay, so I'm going to take you inside the reverse transcriptase from HIV. Here it is. As you can see, it's showing a quaternary structure. There's a number of subunits. It's got different functions. It's got a nuclease function and a polymerase function. And as you can see, look, you've got the strand of nucleic acid running through the middle there. Okay, and you've got your polymerase over here, nuclease activity over there. You don't need to know the structure and the different subunits. You just need to know what reverse transcriptase is and what it does and how we've hijacked that and used that in the lab for our own purposes. Okay, so whether you're doing that, okay, or the second method, going straight to the DNA, straight to the genome, you're going to need to cut the DNA and trim it and cut bits out. Particularly if you're going whole genome, you're going to need to cut the genome up, and you're going to need to do that in very, very specific places. So again, nature has provided us with some brilliant molecular scissors. Okay, and these molecular scissors are called restriction enzymes, and they are an absolute bread and butter standard piece of technology used in modern molecular biology. So enzymes that are cut up at specific sites, okay, are called restriction enzymes. They only cut DNA at specific base sequences. So for example, this one here, right, this one, as you can see, look along the top strand, you've got TG, AATTCA, and then coming down to the bottom strand here, reading it the other way, TG, AATTCAG, right? So you can see that area there will have a specific 3D structure. It's actually what's called a palindromic region. It reads the same forwards and backwards. And as you can see, the restriction enzyme here has cut right at the top between the G and the A, and at the bottom between the G and the A. Okay, that is a very specific location, and if that sequence pops up anywhere in the genome, then this particular restriction enzyme will cut there. This one is called ECHO-R1 because of where it's derived from, E. coli restriction enzyme 1. And the suggestion is, why do these things exist in nature? Is because that bacteria, which usually are our sources for restriction enzymes, use them to cut up viral genomes as a kind of self-defense mechanism. Okay, so that's what a restriction enzyme is. There are lots of different ones that cut at lots of different sites, and they are your molecular scissors. Okay, so they also, the keen-eyed of you might have noticed, don't cut straight. They cut this kind of staggered cut, which again is really handy if you're trying to do some recombination, some genetic engineering, because you should be able to work out, here it doesn't take a genius to work out that if you cut one source of DNA and another source of DNA with the same restriction enzyme, you're going to generate these two ends that are complementary. All right, and we call those sticky ends. Okay, so a sticky end is a short stretch of single-stranded DNA with a complementary sequence, and they're really, really useful. Okay, because it means that when we mix our two samples of DNA together, our for a sequence we want to include and our vector, right, then we basically get complementary base pairing, and that allows us then to start gluing them together. But common exam mistake here, same mistake that gets made in year 12 at AS level, please, please, please don't say there's an enzyme involved in this. It's just hydrogen bonds forming. This is entirely passive. There is no enzyme involved in this. There is an enzyme involved because, of course, just forming the hydrogen bonds doesn't do up the backbones. So we need an enzyme to do that, which we'll come to in a minute. Okay? So what are restriction enzymes for? Well, they cut DNA in precise places. They're kind of molecular scissors. Please don't write that in an exam, but that's what they are. These lengths of DNA cut out are called restriction fragments, and they can be used for all sorts of things, not just genetic engineering, but also mapping genes, because certain people's DNA with certain sequences will cut up in certain specific patterns, allowing you to tell the difference between different families or different species or whatever it might be. Okay, So here are some examples. Echo R1 is from E. coli strain R, and HIND3 is from Haemophilus influenzae, and it's D3, right? It's number 3 that was found. Now, those of you who might have spotted as well, and those of you who are arts and crafts fans will know that if you've got scissors, you also need glue, okay? And that's where DNA ligase comes in, okay? Now, to ligate means to stick together. Now, here's the mistake again. This, the hydrogen bonding of those 4 bases, will only happen, right, passively, right? It's only happening passively. There's no enzyme required to do that. It's not necessary. The amount of times people say, oh, covalent bonds are formed between, no, right? This is passive hydrogen bonding process. The bit that requires an enzyme that forms a covalent bond, okay, is the backbone, okay, so it repairs the broken DNA, joining the 2 nucleotides in a DNA strand, okay, so sticky ends annealed by hydrogen bonds, okay, and then it's DNA ligase's job to complete the backbone. And you should know from earlier in the A level that's phosphodiester bonds, okay, phosphodiester bonds joined back together again, okay. So to put that in its fullness, look, we've got cut up DNA up here. Maybe the stuff on the left is from your donor, the stuff from the right is from the recipient. You get complementary base pairing by hydrogen bonding. There's no enzyme involved in this, that's not happened yet. And then down here, we've got a DNA ligase that's actually joining up where those grey squares are, forming that phosphodiester bond. Right, moving on. Last one then, the gene machine. Okay, so the gene machine cuts up, the gene machine is basically manufacturing DNA entirely artificially. Okay, now I've put this word here, right, because I always say it wrong. Right, so this is just a little reminder for me. You can think of it as in a little speech bubble, right? You don't have to know too much about sequencing and synthesis, although there is information in this playlist about those things. But once you know the code in terms of A's and C's and G's, and you've stored that on a computer, it's basically DNA software, right? And you can send that around the world, send that data to anyone with a DNA printer. Okay, and you can design different combinations of genes and things like that, and then just print them out. And a brief version of how that happens is you basically build the DNA in little fragments, right, called oligonucleotides. That's that word there, right, oligonucleotides, and that happens on a dish in lots of different plates. And the most similar thing you will have come across to this is the formation of ELISA assays. Okay, where you stick one base down, you rinse the rest off, you put the next base on you want, phosphodiester bond forms, you rinse the rest off, the next one, until you get these sort of long chains which have overlapping sequences. You then take those all off the plate, you put them together in a dish, okay, and due to a process called polymerase chain reaction or modified polymerase chain reaction, you can then join them together. Now, you haven't done the polymerase chain reaction yet. There is a video in this series on that. See what I was saying in the beginning about every process requiring about 3 other processes that you don't necessarily know yet. So, what is a gene machine? Well, basically, you take your desired sequence, either derived from the amino acids or from somebody else's work or from gene sequencing. Right, you store that digitally. You initially synthesize these little oligonucleotides with overlapping base sequences, and then you join them together later in another stage. Okay, so this is obviously incredible, powerful stuff, because essentially we've reduced DNA to a code, right? To DNA code, and what that means is that we can actually start building machines that have never been seen before in the world. Okay, we can start taking other people's work and saying, oh, I want something that links this enzyme to this pigment to this functionality, and basically build electrical circuits, right, with the same sort of logic gates. If this, do that, then do that, right? And you can build things that have never been built before. And this field is called synthetic biology. And there's an organisation called iGen, the International Genetically Engineered Machine Competition, which basically pits people together with the best ideas. And you can do this quite easily, right? There are students I've known who've come back after their first year of university and said, oh yeah, I'm involved in making some water purification thing or something like that. So a year after watching a video like this or having a lesson like this, they're off doing this stuff. Okay, so it's very much something that is achievable in the near future for people like yourselves watching this. Okay, so it's an incredibly exciting time to be a biologist. All the wonderful things we can do, all the wonderful things we can make, right? Obviously very, very exciting, very dynamic field, okay, which is why I'm so excited to do this playlist for you and to do these videos for you. Okay, so that's the end of our first video in the Biotech Basics playlist. See the other videos that are done for more complicated and more detailed information on things like sequencing, PCR and so on and so forth, and I will see you next time. Thanks very much.